Okay, the first question of last week was uh, what's the nature of this epigenetics? Is this something that study the environment and the relationship between environmental effect and the gene? Uh, Not exactly, but it involves such relationship and inevitably. So hey, geez, this guy. So this epigenetics is definition. By definition, epigenetics is what uh, the change in trait without any DNA sequence changing. And in reality, all those who study the epigenetics have interest in the gene expression level, like how much RNA can be made out of particular. And practically speaking, those who study epigenetics deal with this DNA condensation, chromatin condensation, because this gene expression level is directly related with this how much chromatin condensation is. Like if particular region of the genes, the chromatin is highly condensed, then gene expression level is down, and the vice versa is true. And that's where this connection between environment and this the genes function comes, because a lot of times this environment can affect this the level of this how hard or how soft this chromatin condensation level. So practically, yes, if you study this epigenetics, you somehow deals with this the relationship between environment and the function of the genes. But that is not the real actual reason or the definition or principle of epigenetics is probably what I can say. So yeah, another question regarding this epigenetic. So if someone chemically controls the degree of gene expression, can that person change to have the desired trait? Yes, theoretically it's possible. So using some chemical or some different modes, you can somehow control at your own will, control particular genes expression level, and you can also monitor or control the effect. I like that. Uh, so, uh, do I look like really passionate about science? Oh, so my deception worked. Uh, well, if I, the reason why I like, or another question regarding why did I major in science? The same questions, kind of. Why do I love science or why I major in science? Actually, to tell you the truth, when I was in high school, my favorite subject was not science. I really, really, uh, I shouldn't say favorite. I did very well. I always did excelled very well in some kind of subject like history. History was probably my favorite subject because it was so easy. It was so natural for me, and also Korean. It was also very easy for me to have a high marks always. So mostly all those those 문과, the humanity, social science type subject was so easy for me. And on the other hand, the science was so difficult. And the reason why actually I can tell why I was so poor, suck at science, because to do very well in science, you have to be very diligent. You cannot really do overnight things to have a very good score in the science exam. But those the 문과 subjects, I can actually work on overnight and then have a very good score. Probably that's why I was really, really lazy student. And then, so I initially went on to I majored in college. I studied the economics. So it's not a science. Uh, and then always I didn't really, the MUCA subject was not my real favorite, although I really did well. I really wanted to study this science subject because somehow I thought this is something more important or more valuable. This social science and humanities are something meaningless. That was really, really wrong proposition, of course, I know. But my very young ages, it's not really. So anyway, that's how somehow what some my personal change in courses, naturally I switched into this science. And by now, in the middle of this science, I'm very old now. The reason still I value this particular subject of life science is because, uh, first, the reason why I love science is something always I can discover new things. I don't have to stick with the old agenda or doctrine or whatever, but always try to discover, find the new thing is something that really excites me. Another thing is, as a scientist in academia, it's always, my life can be very flexible. I always value freedom. So I can be free. I can work whatever, whenever, wherever I want is another thing that I love. And also, a lot of those scientists are control free. Maybe I can tell you later on. So that's how and why I studied this science and then remain in this business. Why do some people can accept information easily while others take more time to understand you? Think again. It's because your priority always. It's just a matter of different priorities. If you think, if this particular information really, really capture your attention or interest, then you will have a really quicker time to absorb it. The reason why some information takes too long for me, for you or whoever to absorb is probably because you are not really, really interested in. So it's just a matter of different interest. It's not a matter of a different intelligence, don't you think? So everybody has a different the interest, a different agenda. That's at least that's what I believe. So not ready to this lecture, but okay, yeah, this is really, really my nightmare. I have to really apologize for this. I know it is really ridiculously late for returning this result of the grade in your midterm exam. I know, and I really, really, I cannot really sleep these days. But I will. I really planning on to return those grading results to you by sometime next week, so that at least you can have some idea about how the way I graded on your the answers, so that it can be reflected onto your final exam too. That's what I honestly and I'm planning on to. So that's what I do. I'm sorry. Okay. And okay, this is it. Yeah. So I mentioned last week that my kind of a dream is to incorporate this machine so that I don't have to teach new students how to work, how to do this skill, but just tell a robot, hey, do this, do that, and then I can be sitting and then only mentally preparing any kind of a research project is my idea. But someone brought about this. How about instead, like vice versa? Don't you dare think that you are genius, right? I agree. The robot or artificial intelligence can actually do the mental work better than you. I agree, but still, the most of this, probably almost all of these scientists are control freak. Their ego wouldn't really allow. They want to have this control. If instead of artificial intelligence can dictate and tell you what to do in terms of designing and what to pursue, and immediately at least speaking for myself, I would immediately lose an interest in pursuing any particular scientific project. That's how I don't want to actually invite the artificial intelligence help in mental part of this scientific performance is probably my answer. But maybe I'm only speaking for myself, but I believe that everybody else, like other scientists, would share the same feeling as mine. That's it. So we can quickly jump on to the rest of the lecture so that we can say goodbye while still the sun is on. Let's see. Okay, so these are three different examples of a particular mutation, and then probably have some compare and contrast of each different cases as a real life scenario or example of a mutation. The first one is sickle cell anemia, which has been introduced before during this lecture already. So, one statement, the sickle cell anemia, what's the cause? Once again, this was caused by a single base substitution. So this is something paradoxical in that sickle cell anemia, we all know that it is really the big thing. The consequence is very serious. Those patients suffering this sickle cell anemia cannot really have a normal function in their life of viability. But on the other hand, it is caused by base substitution. Like 10 minutes or 15 minutes ago, what did I say? Usually substitution, single nucleotide, base substitution is really benign. You wouldn't really have any, the chances of having any impact is very low. So, in other words, this sickle cell anemia, which is caused by a single nucleotide, what I mean by base, single nucleotide substitution is really unusually, extremely unfortunate case. Substitution and somehow, that substitution was disastrous. So, nucleotide, somehow A was converted into T. It's no big deal, seemingly. But because of that, total structure of this protein has been changed. So this is kind of model structure of hemoglobin. And two identical yellow chain and two identical gray chain combined together to make entire fully functional hemoglobin protein whose function is to carry oxygen, right? And there is one particular tiny change of amino acid at a particular position and then entirely disrupt the overall structure, shape of this hemoglobin protein, and that's how. So if you have this incorrectly made, shaped, or folded hemoglobin protein, they accumulate in the red blood cell instead of each different hemoglobin protein floating around in the blood and carry the oxygen, but they combine together to have a really long thread-like, fiber-like shape, and then settle down, then sink in your cell. Okay. So, and as a consequence, your red blood cell cannot function normally. And in the early part, I actually explained that the mechanism why they have this accumulation and then sink. And it's because the change, the identity of amino acid was like, initially hydrophilic amino acid was changed into hydrophobic amino acid. You don't really have to remind yourself once again because it's not going to be part of your final exam, but it was part of your midterm exam. The reason why this thing is because of this hydrophobic interaction was usually this hydrophobic part of a protein will hide inside, fold into. But that particular position, the hydrophilic amino acid was on the surface and then was somehow switched, changed into hydrophobic one, and this hydrophobic amino acid on the surface all of a sudden, exposed. And it's got panic. What am I going to do? I have to go inside. But there is no chance because you are exposed on top of this. So, the only other viable alternative you can make is this. Try to hide this hydrophobic, but each hemoglobin protein combine together to have long things so that they can minimize the exposing this hydrophobic part. And that's the reason why whole thing accumulate and sink. Okay? So this normal versus clumped hemoglobin polymer is like big difference in appearance. So it results in the sickle shaped instead of the roundish red blood cell, but it has a sickle shaped red blood cell. Hey, behave yourself. No, you're not interested in seeing you. Oops. The only difference is red blood cell and the sickle cell. Red blood cell, like that. But the thing is, the function of this hemoglobin is to provide oxygen to every different part of your body. So this defect of ability of carrying oxygen in terms of through this protein can have so many diverse consequences. Think about it. The blood supply is so critical in many different functions of yourself. So it's not only one single disease like you have this lack of the blood supply. That's not it. But you have so many diverse, different, multiple disease symptoms also result due to this defect of blood supply, circulatory system. So this type of example in genetics, you have only single gene, the mutation in a single gene, but the consequence is not one trait, but so many different traits. Multiple traits have been affected by single gene mutation. We call this as a what? Pleiotropy. That's a pleiotropy. So when someone refer, hey, a certain gene mutation has a pleiotrophic effect, then that's what it is. And if here the point is, yeah, in genetics, if you simply just listen to the genetic lecture, sometimes lecturer will explain, okay, this gene will have a pleiotrophic effect or something, and then you have a tendency to mistakenly try to, without thinking, try to memorize, okay, this particular gene's mutation has a pleiotrophic effect. Okay. But if you think about why any particular gene mutation will have, on some occasion, have a pleiotrophic effect, then you can easily actually predict, If you have this mutation, then probably you will have a pleiotropic effect, because this is an example. If you have a defect in the blood supply, what's going to happen? If you have a defect in the blood supply, then probably you will have a defect in the oxygen supply. Okay? And then if you have a shortage in the oxygen supply, then you will have so many different consequences. Okay? Because oxygen is so vital in every, because oxygen will determine how much ATP, how much energy you will have. So you have so many different areas or so many different functions that is going to be affected by a single gene defect. No wonder. So many cases, in many, almost all occasions, you can actually, without even knowing it, you can predict, okay, if this gene's function is disrupted, then probably it will be a pleiotropic effect, okay, without memorizing it, is my point. So, yeah, look at this. This single sickle cell anemia disease, physical weakness, obviously, yeah. If you don't have enough oxygen, then you will feel weak because you don't get enough energy. Right? And anemia, 빈혈, anemia, of course, yeah, you lack of this oxygen is equal to the short of this blood supply. And heart failure, obviously, to have a healthy heart function, you have enough energy supply is vital. Right? And pain and fever, obviously, yeah. And brain damage, brain requires so much supply of oxygen. And obviously, if you don't have enough oxygen supply, your brain will be easily damaged. So that's why so many different such effects will come from a single gene mutation. Do we really need to memorize all these things? Obviously not. There's no point. So that's the pleiotropy. But there is another. Some occasions, so this is pleiotropy, but opposite. Sometimes you have only single character changes, single character change in trait, but that is contributed from multiple genes. Okay. Multiple genes working together and only pointing to one particular trait. Human height is a thing. So many multiple genes work together to determine human height. Human intelligence. So many different multiple genes work together so it will be very difficult to, if you even know first of all, that's why you cannot really pinpoint what kind of gene control your intelligence. If that was determined by a single gene, probably already some weird scientists would have attempted to change the thing to their advantage to artificially increase the intelligence. But it's very difficult because thanks to this whole thing, it's called the polygenic inheritance. So intelligence is another such good example of. Another thing is human skin color. This human skin color also is determined by not a single, but many interaction of so many different genes. So there are so many traits that actually are determined by many genes. Another characteristic of this sickle cell anemia is it is a recessive mutation. And as I explained earlier the other day, we can easily estimate or predict that this sickle cell anemia would be dominant or recessive mutation, because we knew the molecular nature of this mutation, which is what? Due to this substitution normal hemoglobin function was disrupted. So in other words, the normal function was lost. Whenever you have a loss of a function, this mutation will confer what? Recessive. Why? Because existing normal function can always cover up this. So if you know what happens, then we can predict whether it will be recessive or dominant. And in the case of this sickle cell anemia, we can correctly predict it will be recessive because simply due to this mutation, it cannot function anymore, lost. In other words, if you luckily have another, because we are luckily diploid, so we have another copy of gene exists, and if this gene is still working, then this working copy can cover this lost function. Okay. However, so in other words, usually even if you have this mutation, heterozygous, having one normal copy, although I hate saying normal, but one non-mutant copy and this mutant copy, then physically, in appearance, they will be, so to speak, normal. Their function is not going to be really challenging. However, it's a totally different story that if you have this mutant, then you will have extremely, extremely difficult situation to be able to survive because, how can you survive even like this lack of this supply of oxygen? It will be really tough for you. However, this weird thing is heterozygous has a strange advantage against this particular type, malaria infection. And nowadays, actually, the actual molecular mechanism has been uncovered. So we know why this heterozygous individual can have a somehow selective advantage, which I wouldn't really share with you the reason. Because previous several years ago, I did actually during this classroom, I introduced actual molecular mechanism why this heterozygous has a selective advantage. But why? Why do you have to really worry about so much detail? So just this is good enough. Heterozygous individual have for some reason they have a stronger resistance. They have a stronger resistance against this malaria infection. They can live better, survive better. And that is the reason why this bad gene, this sickle cell anemia gene, was not entirely kicked out of the population. Because whenever you think about it, during our successive reproduction and producing offspring, some combination of offspring somehow result in this two mutant copy, which means the homozygous will result in. Then what happens? This individual will die out. Okay? This individual will have a difficulty in producing offspring, right? But still, somehow, the amount of this bad gene in the population remain somehow constant. Why? Because of this surprising advantage of this at least in the heterozygous status, you can survive better. Particularly where? In the area where this malaria infection is problematic, like on some certain area of Africa in particular. So it's no wonder why if you examine the population, especially in the Africa, the population, the frequency, human living in the Africa, carrying this mutation in the population is very high because of this advantage. If you carry at least one copy of this mutant, your survivability is there. That area is better. So, okay. So that's the area where mostly this trait, sickle cell trait, so distributed in high. The pink and red areas are where you can find the most frequently. this trait, and it will match where this malaria endemic area kind of closely overlap with this. I don't have to go over this thing again once again because why this risk over the wild type allele? Why is the sickle cell allele recessive over the wild type allele? Because the mutation, the nature of the mutation is loss of a function. That's why. And why some mutant allele dominant over the wild types? Why? Because in that occasion, this mutation is something like result in the gaining of a new function, or at least somehow disrupting the existing function. This mutation will actually interfere with the new existing function, or sometimes totally different function, new function. In that case, existing function cannot touch this thing, cannot have any control, and then it will show off. That's the definition of a dominant. Now, the second example of this mutation is a cystic fibrosis. This one is not a substitution, but deletion. But luckily, seemingly luckily, this cystic fibrosis mutation result in the deletion of 3 base nucleotide, or maybe should I say caused by 3 nucleotide deletion. So would it disrupt the reading frame, like frame shift or not? No, because 3 nucleotide, so it will preserve the reading frame of a protein. So what's the problem? What did I say? So now today I'm revealing what a liar I am. Everything I say is a totally lie. Something like 20 minutes ago I said the 3 nucleotide case level change, like a 3 nucleotide deletion or insertion, will have usually no effect at all because you will get to preserve reading frame. So this is perfectly, exactly that is the case, that cystic fibrosis, you lost 3 nucleotides at a time, so you get to preserve your reading frame. No consequence should occur, but serious consequence occur. So this is another example of very unlucky, unfortunate incidence of even with this 3 base deletion, you suffer really serious consequences. This, once again, this is another example of what? Pleiotrophy. So many different, oh, wow. Yeah, if you're planning on entering the medical school, probably you might start trying to memorize all of this. If not, probably forget it. This just only suffice to know that, whatever it is, the cystic fibrosis mutation will be also something can be regarded as a pleiotrophy. And this is something, mutation on the gene coding for this protein called the CFTR, whatever it is, CFTR protein. The full name of a CFTR protein is a Cystic Fibrosis Transmembrane Conductance Regulator, whatever. So this is membrane chain protein, membrane-bound protein, and it's ion transporter membrane protein, transporter protein on this our cell membrane. And we usually describe this mutation as is like delta F508. Not that is important, but something like if you find sometimes that cystic fibrosis is like this delta F508. What does that mean? This delta stands for D, deletion, indicated deletion, deleted. F is a particular amino acid, phenylalanine, one letter word. So 508 is this is very big protein. So this particular phenylalanine happens to be present on the 508th position of the entire which is much like several longer than 1,000 amino acid protein, and it's a 508th position of particular amino acid, which is a phenylalanine, has been deleted out, missing entirely. So this is only one single amino acid is gone. Everybody else is perfectly fine. So usually you can expect that this function, this mutant protein should also function normally. But no, in this case. So this function as an ion transporter and is shaped like this. And bottom line is, what it does is it maintains the flow of water outside of our lining of the our esophagus or the respiratory the channels. So what happened is that protein folding was disrupted because of this missing of one amino acid. Entirely, this protein is misfolded. Okay? And if you recall, what happens in the rough endoplasmic reticulum, inside of the rough endoplasmic reticulum, the two things happen. All those, the secreted protein, including the membrane proteins, are all enter this endoplasmic reticulum, okay, rough endoplasmic reticulum. And what happened to them? One is protein folding. And the other thing is glycosylation. Carbohydrates are added. And this folding is very important in that if you fail in this folding, then that particular protein's functionality is not guaranteed. That the function will be lost. So cells consider this folding very seriously and then monitor very tightly. So if any protein fail in proper folding and they're not allowed to go out of this endoplasmic reticulum, they are destroyed on site, they are degraded. And that's what happened in this case of a CFTR mutant protein, cystic fibrosis protein mutant. They, because of this absence of this particular amino acid on that position, they have a difficulty in folding properly. So they fail miserably in presenting themselves, oh, I exist in the properly folded structure. And the cell noticed this, and all these misfolded proteins are actively destroyed inside. So no protein, no CFTR protein can be available on the cell membrane if this mutant, the individual having this mutant happens to be what? The homozygous mutant. If they are luckily heterozygous, still one normal copy of gene can produce normally folded protein, so those proteins can be present on the cell membrane. So once again, without going any further, we can predict the consequence of this. Would this be recessive or dominant mutation? What do you think? Will it be recessive or dominant? Mutant function will be what? Simply lost. So if you have a normal copy of a gene, still functional copy can be presented. So it will be recessive. Yeah, once again, it will be recessive, for example. And what happens is, especially if you the homozygous, in other words, you don't have any normal copy of this gene. Both of these genes are mutant copy in terms of this CFTR gene. Then you have this problem of building up of thick mucus on the surface of the lung and this esophagus. So because of this very thick mucus area, it can easily trap all those incoming infectious bacteria and viruses. So you are more vulnerable to any various infection. That's why you have so many, the multiple effects of pleiotropic effects out of this. The thing is, normal job of the CFTR protein is transport particular ions in and out. And this in and out is very important in that ensuring proper flow of liquid water outside of the cell. But if you don't have a functioning copy of this CFTR gene, then this water flow cannot be efficient. So, like, think of this: the river have a normal flow of water, do not have any really undesirable things floating or sinking. But if you have an unflowing, the really slow moving water or pond, you have so many developed things, and then the water becomes so dirty. And similarly, because of this, you have a very thick mucus buildup, and to which so many different infectious microorganisms can settle in, and then they can have a much easier chance to get inside. This is another such diagram illustrating the difference between this normal functioning CFTR versus malfunctioning CFTR. And as we went over, this will be another recessive mutation. And the reason why we get to see so many times this particular incidence of CFTR, although the Asian population, the CFTR, the mutant frequencies are so rare. So in our neighbor, if you look around, you're like family and friends, and probably you hardly get to hear about anybody related suffering from this CFTR in this particular region. However, the European population, the story is different in that they have an extremely high frequency of this particular lineage, genetic lineage of the European population. So it's even in 1 in 3,000 homozygous, I'm talking about, not the carrier, but the homozygous mutant individual occurrence, the rate, frequency is even in 1 in 3,000 is just such a lot. So they take this so seriously and then introduce this instance in the biology textbook. And unfortunately, those textbook and the knowledge was all founded by those European heritages. They were the front runner of this whole the science. That's why we get to hear a lot about this. The last thing is lactase persistence. Is another mutation. Okay. And this incidence now is caused by another base substitution. Wow, okay. So if that was a base substitution, probably one base substitution, so its chances are that you will have what? Frame shift, single base, substitution. So chances are you're not going to have any effect, right? But yes, this is a very interesting case of mutation that so if you have this mutation, you actually can enjoy much longer of this one of my favorite food substances. It's like ice cream or dairy products. I love ice cream, but humans are programmed to stay away from this dairy product as we grow older. Genetically, we are programmed. So like, for example, normally programmed individuals at the age of old age, like your 30s, 40, or 50s. Now it's not like the old times that whenever you have this dairy product, you have like the difficulty in your stomach and the intestine, gas buildup. So that's exactly what happened to my wife case. When we were 20s, we were together, like we stayed up all night, emptying whole bucket of ice cream together and then chatting. So we had no problem. Okay. Now, this age, I still don't have any problem. So I can eat whole elephants of ice cream overnight, but no longer. My wife can. So whenever she has a small amount of ice cream or any dairy product, she has a little difficulty in this. So we cannot really have a really precious moment sharing together over the ice cream. That's a what a shame. But that's what happened. That's what you are supposed to be, because this dairy product, like the milk product, this is caused by this lactase intolerance. Is in order to handle properly this dairy product, you should be able to digest the milk sugar, lactose. And here, the enzyme that can digest this milk sugar, lactose, is called lactase. And the expression of lactase is only programmed to be high and active when you are baby, infant. So, sounds reasonable. When you're a baby, you only get to rely on your mother's milk. Then when you grow older, you no longer need any mother's help for your food supply. You can find something else, like alternative or better food source. So why do you still have to spend your energy on producing this lactase enzyme? So that's how you were originally programmed. So the expression level of this lactase enzyme gene is supposed to be dwindle, okay, as you grow older. And somehow mutation occur, especially in some European population. Somehow mutation occurs. So they can enjoy the lactose digesting ability persistently over long age. That's what is called lactase persistence. It is a mutation, but it's a pleasant mutation. I love it. So that's what I said. Some people can have a sustained expression of this lactase, like me. And now, the nature of mutation was discovered eventually, like after completing the Human Genome Project. And this base substitution mutation was actually located on the far distantly away from the lactase gene coding region. Several thousand nucleotide base away from actual gene. And this enhancer region is something that serving the DNA sequence, serving for recruiting the transcription factor. So this is something like a binding site for the transcription factor for some reason, whatever it is. So the bottom line is, this mutation is not on the coding area, if you still remember what the coding region was. Coding region of this lactase gene itself, but the controlling element distantly located, far away from this gene. And this is on one single mutation. Nobody can even notice this difference, because it is not even on the coding area. But that's still minute change affect really differently about the expression level of this. Not even 14,000 nucleotide away from actual lactase gene. If there is a lactase gene, 14,000 gene away from this thing, only C to T change was the thing that responsible for. This is very interesting thing. Wow. How can that be? Although at this point, the kind of amount of information that I provided cannot really enoughly explain the reason why this relationship. To do so, I have to provide some more detailed information about this gene expression control, but yeah, I'm not going to do that. But simply, the only important thing is this mutation is something that located on the control element, control region of this actual coding region. And another thing is now, would this be a dominant or a recessive mutation? Let me ask you. That's all. What do you think? Dominant. Why? Yeah, you're correct. But why? It's a function, new function. So as a consequence of mutation, you gain the new function. So the existing function cannot do anything about. So this is why it is regarded as a dominant mutation. Right. And so a lot of those Caucasian population carry this mutation. So that's why they have no problem, and they have such an intimacy with those different dairy products. But unlike, on the other hand, those East Asians and Native Americans have a very, very low frequency of the mutation. So that's why they have especially like a certain particular difficulty in this, at their old ages consuming this dairy product. But what about Mongolians? Mongolians actually depend heavily on those milks, sheep milks, and all different dairy products that they have to rely on their main food supply. And as a Mongolian, just so they genetically should be very close to the Eastern Asians, like Koreans and Japanese. Well, what happened to them? Do they carry unusually high frequency of this mutation? What do you think? It turned out to be quite interesting in that, genetically speaking, they were very similar, close to other Eastern Asians. So they don't carry these mutants, the lactase persistent mutation, but still they are able to handle this dairy product. How? What do you think? What magic do you think they were able to play? Microorganism. Their guts, microorganisms, they somehow invite all different microorganisms. They can do this digestion. So once again, revealing the importance of this microbiome, microorganism living inside of things can make such differences. So I think that's all. So, okay. So all these lectures are gone, and as I announced briefly in the beginning of the lectures, the exam question will be uploaded like as late as next Friday. But once again, like I said, it will be high chance of me doing so a little earlier, like a couple of days earlier. Whatever the case, you will have more than 1 week to submit the answers, just like the case of midterm. So good luck and have a nice summer break.